Good afternoon, everybody. We're going to go ahead and get going. We're going to discuss the role of developmental genes, at least in a very introductory way, in body plans or body designs, which means we'll be talking about symmetry a little bit more. We'll discuss sponges and then go ahead and move into the protostomes. One announcement, I have updated the not responsible for document, which is a short description of if there's parts of the chapters that you're not responsible for, then I make those explicit as possible to try to help you. Now that we're marching through the different texts, of course, it's the key lineages that are the main thing. So I've tried to be really clear about which key lineages you are and are not responsible for. I know it can be very overwhelming with a very large number of key lineages, so we're going to try to keep it tight so that you understand the major different animal groups without getting totally swamped in details. Let me know if you have any questions about that. I showed you this figure before. This figure, which is not from your textbook, is a diagrammatic representation of different kinds of symmetry. And when we're talking about animals, they are usually broken down into three major groups of simplicity. Radial symmetry, which is shown up here at the top, bilateral symmetry, or no symmetry whatsoever. And again, we've got a living creature on the left, we've got a sea anemone shown on the top, and a lobster on the bottom, and then over on the right-hand side, we've got inanimate objects to try to help make it use more familiar objects, hopefully, for explaining these concepts. One of the things I like about this diagram, which is why I'm showing it to you, is that the mirrors, the planes of symmetry, which you can think of as a mirror as reflecting the two halves, which are identical to each other, are completely explicit in this diagram, which I think is terrific. What is maybe less obvious is, of course, in radial symmetry, there's not just two planes, but an infinite number of planes. You can think of any kind of rotation of the plane around the longitudinal axis, and it would be a plane of symmetry where there are mirror images on each side. And by the way, this has absolutely nothing whatsoever to do with the orientation of a body as a whole. If we take the flower pot and the sea anemone and turn them sideways, it's exactly the same argument will be made, is that you can rotate around the longitudinal axis, and the one side is always reflecting the other. Now, of course, the big deal is not so much the type of symmetry or geometry, it's the association with all the other changes in the animal lineages that are considered the bilaterians, those who are primarily associated with bilateral symmetry, and those are cephalization, right, the concentration of the nervous system and sensory organs in one end of the organism, and also the existence of a special kind of body cavity which is surrounded by mesoderm and is formed during the embryonic stage. So these are some of the other important phenomena that are linked to bilateral symmetry, even though hypothetically one could, of course, imagine that they wouldn't have to be linked. They just are within the animals. If that makes sense. So let's test your symmetry here. Make sure that you understand what's meant by symmetry. We're going to start with some inanimate objects. Here's one. It's a stop sign. So consider, the stop sign is a hole here. Okay, ignore the trees and the sidewalks. I want to know, how would you best describe the symmetry of this object, the stop sign? It's circled in red here, making parallels with how we talk about animal symmetry. Radial, bilateral, neither. Chat with your neighbor. 10 seconds should do it. Click in. I'll see what you guys think. Okay, everybody should be clicked in here. to go ahead and stop this. Let's see what you guys think. Let's move this animal. Fantastic. Not all of you, but at least most of you got this one. Yes, absolutely. Oops, wait a minute. Yeah, right, bilateral. If it was, why is this not correct? Because you can't rotate it around. It's not like a lollipop. It's flat, right? The key of something that's bilaterally symmetrical is that we end up with a left and a right side, as well as a back and a front. So let's try another one. We're on a roll here. Let's try another inanimate object here. A pencil. What do you think? Radial, bilateral, neither. You can take 10 seconds or so to formulate an opinion and click in. Already clicked in. All right. Let's see here. Fantastic. Yes, radial would be the preferred answer here. I'm actually surprised how many of you picked bilateral for that, right? Radial meaning you can rotate around here. There's not really a left and a right side of a pencil, right? If you're distracted by things such as, you know, there's writing on the side of the pencil describing it as a number 2 pencil or not, that would be considered a relatively minor thing that wouldn't cause one to reject radial symmetry. Okay, let's try an organism now. T-Rex, what do you think? Radial, bilateral? Okay, I'm going to close this down. Fantastic. Best one yet, absolutely. Good job. We're going to do one last one here. Let's try this one. An earthworm. Okay, again, just based on the symmetry, what you can tell looking at it, what do you think? Radial, bilateral, not really either. What do you think? Okay, I'll go ahead and close this. See what you guys think. Fantastic. All right, good job. Okay, I'm surprised, honestly, it wasn't 100%. So hopefully those of you who got it right were recognizing that if you rotate this animal around, looks pretty much the same. Yes, there's some tiny differences. One of the things that makes this specific example a little bit more complicated, this is an earthworm. If those of you who know something about invertebrates may have recognized that this is in phylum Annelida, which is absolutely a member of the Biolateria. So technically, earthworms are considered a member of Biolateria. So in terms of being a member of that group, they are absolutely in that group. No ambiguity about it. But if you look at the body by itself, is it bilaterally symmetrical? Well, no, not really. And in fact, many of the worms are not, especially if they're cylindrical like this one. So I bring that up to emphasize to you that the overall body shape and whether or not there is radial or bilateral symmetry is not a completely unambiguous identifier for position in a phylogenetic tree such as this one. So this phylogenetic tree of evolving animals, this is figure 33-2 in front of you right now, and does show the what group is the Bilateria, right? So it includes all of these organisms, almost all of them, in fact, as shown here. So why the term Bilateria was chosen to include this group instead of something else like Cephaloteria or Cephalosalomateria or something? You know, if I was in charge of making it up, I would have picked something like that, because in fact, this is somewhat confusing when one does have things like the annelids and other kinds of worms, which if you just look at them, then they do appear to just have radial symmetry. So again, it's not the bilateral or radial symmetry per se that is really important about the Bilateria, members of the Bilateria, the Protostomes and the Deuterostomes. Rather, it is all the other associated aspects of body plan, the concentration of the sensory system at the head end, usually forming a brain, and the existence of a very special kind of body cavity. So when we're talking about symmetry, then we're using terms that relate to body axes, so that might be helpful to go through that explicitly. This is actually a figure from a different chapter in your textbook. It is from your textbook. It's figure 22-3, so you can access this electronically, even if it's not in your hard copy, because that's not a chapter that we have bound in the hard paper copy. This shows how body axes are defined in organisms which are in the Bilateria, and the fact that these body axes are established usually in the embryonic stage. So we've got an example of a non-embryo at the top. We've got a little girl, picture of a little girl with the axes marked, and then down below we've got a embryo. It's a mammalian embryo. It's actually not a human, it's a mouse, but it'll look very similar. So if we consider the axes then here, let's go through these one by one. So the anterior-posterior axis is shown here in yellow. So it's towards the head and away from the head. So it's this double-headed yellow arrow, which indicates one axis within the body of animals that are in the Bilateria. And this is set up in the embryo, and you can tell because of what's developing where within the embryo. So there's that one axis. And then perpendicular to this is a second axis. Let me switch to the colors so I can match. Ventral dorsal, so that means towards the back, which is the so-called dorsal surface, or the ventral surface, which is the front or the belly surface. So this is at a right angle then to the anterior-posterior or head away from head axis, and same in the embryo. Now the presence of these two axes, anterior-posterior and ventral dorsal, the two of them combined then set up a right-left. So this is shown in this particular diagram as an orangey kind of color, but I think if you think about this in sort of the 3D context, then it probably is obvious to you that if you have anterior-posterior differences and dorsal-ventral differences, then you end up with a left and a right without having that as an additional axis that has to be necessarily identified with its own differences. And in fact, in bilateral organisms, these are the two that are mirror images of each other, right? Are the right and left, as defined. Now, in addition to these basic body axes, there's also words that are used to describe the orientation with an appendage. So we can add that as a, let's see, I want to get a different color, maybe bright red, and appendage axes. The little girl has an arm sticking out, which is very convenient because that then allows us to draw a proximal distal axis. So this is something that's relevant to any kind of appendage that's sticking out for the body. Proximal means closer or proximate. Distal means farther away or more distant. So this is then an appendage axis terms. Okay. So these terms are used in the chapter, so I thought I'd go ahead and explain them so that you know how to interpret them as you're reading about them. And these are established in the embryo by a variety of different kinds of developmental genes, which we will just touch on very briefly. The Hox genes are a set of genes that you probably have been learned about a little bit before. You certainly had a figure earlier this quarter that showed the distribution of some Hox genes within the animals. This is figure 28.2, and you saw this figure before in the context of thinking about homology, meaning similarity between organisms that is caused by shared ancestry. So in this particular phylogenetic tree from figure 28.2, then we've got the sponges up at the very top, and they're labeled Hox-like genes, but no Hox genes. So in other words, these genes are similar enough that they can be identified as being really quite closely related to the Hox genes proper, but the Hox genes, as defined by their role in development, then are actually only found in the animals that don't include the sponges as shown here. So Hox genes have undergone replication or duplication, and so they are homologous to each other in the sense that their similarities between Hox genes in a single individual are caused by shared ancestry at the gene level. But in addition, if you compare organisms, such as was done for your convenience, comparing the arthropods and the chordates in this particular phylogenetic tree, then you can see from the color coding that there are certain Hox genes that can be associated between the two different taxa, and so these are considered homologous to each other in a different kind of way. So these genes then are primarily responsible for controlling development in the anterior-posterior direction. So let's go ahead and go back to this figure. And let's see what color do I have? Red? Okay, I'll go back to a darker color. So anterior-posterior, this is set up by Hox genes. And to a certain extent, also proximal distal differences, but Hox genes are responsible for generating differences in the different parts of the body along the anterior-posterior direction. And in the ventral dorsal, let me switch to light blue so I can make it consistent in terms of color. There we go. So ventral dorsal, there is a different gene involved discussed in your book, and that's the DPP. We'll go back to that in just a minute. Okay, so those are the genes that are involved, some of the many genes or gene types or gene complexes that are involved in setting up the axes within developing embryos and within humans. So how does this work in practice then? These are just colored rectangles. If you look at their distribution in developing embryos, here's an example of an arthropod on the left here, and then a chordate on the right. So we've got a fly maggot, which is a fly embryo over here on the left, and a mouse embryo is an example of a chordate over here on the right. And the color coding in the body parts shows the gene expression of the different Hox genes along the anterior-posterior axis. So in both of these cases, the head is on the left end. Let me grab a different color so it might be less confusing here. So anterior is in this direction in both cases, and you can see that there are differences in gene expression of which Hox genes are being expressed in the different parts of the body along the anterior-posterior axes. So in addition to the Hox genes being tremendously important in directing development, your textbook went through an interesting discussion of the role of the so-called DPP genes, decapentaplegic genes in animals, and specifically they discussed an experiment where the scientists were interested in evaluating their activity in non-bilaterian animals because, of course, when we're talking about all those body axes, we're primarily concerned with bilaterian animals. And so the interest there is at what point in the evolution, in the phylogenetic tree or during the evolutionary history of animals did these different kinds of developmental genes start directing the axes and directing the overall body plans for these different animal groups. So in this experiment then, they used an example of a non-bilaterian animal. Specifically, they looked at a cnidarian, and this is a picture of the cnidarian larva that they used. And they looked for the pattern of expression of these two different kinds of genes. And of course, if if they weren't expressed at all, or if they were expressed everywhere, then that would suggest they were not important in setting up any kind of body axes, even in these non-bilaterian animals. And what they found was that there was, in fact, some pattern that was somewhat consistent with what is seen, although much more simple and rudimentary than what's seen in the bilaterian animals. And that is that, yes, the Hox genes, they showed difference in expression from the what would be the sort of possibly the head equivalent end and the tail equivalent end, or the non-head end in the Hox genes, and also a bit of a dorsal ventral signal in the so-called DPP genes. And so they are seeing then evidence even in the non-bilaterian animals for involvement of these genes. So what is the significance of that? If we go back to the phylogenetic tree of all of the animals, so this is figure 33.2 in front of you right now, what this means is that it wasn't just sort of, one point at which all of these different aspects of the bilaterian body plan developed all together, but rather it evolved fairly gradually, not all at once. Let's see if I get a different color here. What that means is this is something, so the Cnidaria is the kind of the larva that we were just looking at in that experiment is a Cnidarian. So clearly there was a little bit of a hint that along this area here is where the various events occurred that together sum up into what we recognize as the overall bilaterian plan, meaning the 3 different germ layers and the cephalization and the coelom and all that. So. So let's spend a few minutes then talking about sponges. This is a fascinating group because they're so different from the other animals, and of course they're not particularly closely related to the rest of the other animals. And I'm going to go ahead and start out with a wonderful question that was posted on Piazza. I grabbed it as a screenshot, and I've got it in front of you right now, along with the relevant figure from your textbook, figure 33.2. And I noticed that James, one of your TAs, already did a very nice, posted a very nice answer to this question, but I'm going to go ahead and answer this with a little bit of a visual backdrop as well. So the question from the student regarded the paraphyly of sponges. And so the student asked, what does the paraphyly of sponges have to do with supporting the hypothesis that the common ancestor of all animals was sponge-like versus the idea of sponges being a monophyletic group? The textbook says that if sponges are monophyletic, the distinguishing characters could have evolved later rather than in the common ancestor of all animals. Then it went on to give an example of how the common ancestor of humans and chimpanzees was not necessarily chimp-like. Very well put. And so are sponges paraphyletic or not? Well, for a long time it was thought that sponges were monophyletic, but now they are shown as a paraphyletic group as in your textbook. Right here, you can tell they're paraphyletic because here are the 2 branches. It doesn't pass the one SNP test. Here are all the other animals, so I collapsed all the rest of the branches into a single branch here. So this is our best guess at the true evolutionary relationships between the sponges and the rest of the animals, which is that they're paraphyletic. So paraphyletic sponges over here on the left. And we can contrast that with an alternative, which is the choanoflagellates. So over here we'll put if sponges were monophyletic, right? Monophyletic. So there's the choanoflagellates. Unfortunate color choice here. Let me make it a little bit darker. And, oops, there. Sponges, and then all the other animals. Right, so if sponges were monophyletic, we'd have a phylogenetic tree over here on the right. If they're paraphyletic, which most of the evidence points that they are, then we have the scenario over here on the left. So how does that relate then to the question that we're considering? Well, if we consider S as being some sponge-like trait, then, so we'll go ahead and put S as our trait on our sponges then, and we consider the evolution of this trait If one goes back in time, then let's start over here on the more simple monophyletic group, which is on the, or monophyletic representation, which is on the right-hand side. If we consider that sponges have this S, this sponge-like trait, and other animals don't, then if we look at the recent common ancestor, then just based on what the descendant lineages have, there's sort of a 50-50 chance of, what did the common ancestor have. One usually looks to the outgroup for more information on assigning ancestral or derived conditions, assigning polarity to that trait, understanding if this is an ancestral or derived condition. But over here on the right, since we have a paraphyletic situation here, then if we look at this sponge group that is most closely related, that is the sister group to other animals, and we consider whether that most recent common ancestor had the sponge-like trait or not, and we look at the outgroup for these two, then that is in fact another sponge lineage. And so that makes it more likely then, or more probable, that the common ancestor of this group as a whole did in fact have the sponge-like characteristics, would mean that the interpretation would be that S evolved here and then would have been lost in the other lineages. Whatever this sponge trait was that is just peculiar to the sponges is not shared by the other animals. But what a great question. So sponges are actually considered a relatively small animal group, but there are over 8,000 species, and that's 8,000 species that are still alive on the planet now. And some of them are just gorgeous and striking colors, so let's learn a little something about the sponges. If you look at their overall body plan or shape, you can see that some of them do appear to have basic radial symmetry, right? Anything that's cylindrical is basically radially symmetrical, but others are fairly asymmetrical as shown here. And so you see those different kinds of body forms within the sponges. Irregular, and then more or less cylindrical or radial symmetry is shown here. Let's go through some sponge fun facts. They're members of a single phylum, although, of course, scientists are not very enthusiastic about taxonomic units that are paraphyletic. So if, in fact, with more information coming in about the sponges, if they continue to be seen as paraphyletic, we're going to see them broken down into more than one phylum. But at the present time, they are still just grouped into a single phylum, and that is a phylum Porifera. They're sessile as adults, meaning they don't run around. They're stuck in place, meaning attached to the surface, as shown here in the diagram from your book on the right. So down here, this particular sponge is attached to the surface here. Although they're not moving, at least very much, as adults, they can swim as larvae, and so as larvae, the little tiny microscopic larvae are covered with cilia, and they can swim around in the water. All sponges are aquatic, meaning they all live underwater. It can be seawater or freshwater. There are sponges of different kinds, but they are 100% aquatic group. There are no sponges that are terrestrial, and this relates very closely to how they make their living. And of course, they're the sister group or sister groups to all the rest of the animals. And they're relatively simple in their overall body organization, and they don't have germ layers. They lack true tissues and organs. So the cell layers on the outside are not considered epithelia, because an epithelium would be considered a true tissue. So very, very different from the other organisms. They make their living in a way that's completely the same as the choanoflagellates, meaning that they filter out organic material from the surrounding water. And so again, this is related to their aquatic lifestyle. There's just not enough particulate organic matter in the air to have a filter feeding or suspension feeding lifestyle as a terrestrial system. All filter feeders and suspension feeders are always aquatic. So, and just like the choanoflagellates, of course, they also have very similar cells that do the generation of the current in the first place. So the inner lining of the major opening here of the body has a number of different cells in it that are dead ringers for choanoflagellates, although they're in fact animal cells, not protist cells, these choanocytes that have the flagella that beat, and that's what draws the water through. So I think it's a little hard to understand the overall body design of a sponge because it's just so different. You basically have a perforated hollow bag where the wall of the bag is the actual cells that make up the sponge. And so if this is a marine organism, then it would be in the seawater, and seawater would be moving through the sponge, entering this space right here and exiting through other holes. So this part here that is called the interior is not considered a body cavity, so it absolutely has nothing in common with, say, a coelom or other kinds of proper body cavities, which are completely enclosed within the body, meaning they're surrounded by tissue. This interior right here is completely open to the outside world. So all of the cells of a sponge are pretty close to the external environment, the seawater or the freshwater, depending if they're marine or freshwater. And most sponges are in fact hermaphrodites, meaning that they are capable of producing both sperm and eggs. And so what happens is that the male part of the sponge then produces sperm, and sperm are released along with the water that's leaving the sponge and enters the surrounding water. And then how does it ever meet an egg? Well, the eggs are actually retained in the sponges, and so as water is drawn into the sponge as part of feeding, then sperm can enter along with the feeding currents, and if it's the right species, and if the sponge that's entering has eggs that are at the right level of development, then the eggs can be fertilized. You end up with a zygote, it develops into a larva. The larvae are ciliated, so they swim away. They just swim out of the sponge, swim away a bit, and then land, and then they take up a sessile or attached existence wherever they happen to land. So that's the sponge life. And just like plants, which of course are attached in place as well, you might consider that being attached and not being able to move can be very hazardous, meaning you can't get away from something that is trying to eat you or consume you. So just like plants are extremely well chemically and physically defended, so are sponges. And so chemically defended meaning that they synthesize a whole host of different kinds of chemical compounds. Some of these are toxic, some of these are just distasteful, and so they prevent other organisms from just gobbling them down, because after all, they're just animals there on the seafloor. It should be a handy meal. Notice that humans, for example, do not eat sponges either, and it's because they're really quite unpalatable. And so they make such extraordinary chemical compounds, by the way. The pharmaceutical industries are very interested in analyzing sponges for the different kinds of compounds that they synthesize. Some, for example, are found to be very powerful antibiotics, because of course they're also dealing with having to prevent microbial growth on their own surfaces as well. So that's chemically defended. Physically defended means things like hard tissues that make it difficult to eat. And so in fact, within the skeleton body, and I've grabbed a figure from elsewhere, this is not a figure from your textbook, because the figure from your textbook didn't show the existence of the little items that are embedded in the body wall. They're synthesized by the sponge cells. These little spicules are very hard. They can be made of silica, or they can be made of calcium salts, but they make it very, very difficult to consume the tissue by ingestion or eating the sponges. So let's turn our attention now to the protostomes, one of the two groups within the Bilateria, right? There's the protostomes and the deuterostomes. So where does the name come from? Hopefully you remember where the name comes from, and that is from what occurs in the embryo, right? This is figure 3312 from your textbook, and again, just to remind you, during gastrulation, so you're looking at a gastrula, that embryonic stage right in front of you. the invagination, this opening here, the pore, which is called the blastopore, it becomes the mouth in the protostomes, and this is one of the key characteristics of the protostomes, and that's where the name comes from. Proto meaning first, stome meaning mouth. So the first opening becomes the mouth. And another characteristic of protostomes, again, another embryological one, is how the coelom forms within the mesoderm of the animal. But these are embryological features and are not things that you can see actually looking at the adult animals. Protostomes are not obscure animals. Most of the animals on the planet are in fact protostomes. This is figure 34-1 from your textbook, and it is a pie chart of all the animal species. Every single animal species is shown in a pie chart representation, meaning that if half the species of one particular group, then that group would get half the pie in terms of the overall area. So if you look at the legend here, you can see that the protostome phyla are shown in bright colors. Not all the protostome phyla are shown in here, just simply because some of them are so small that if they were drawn on here, they wouldn't even show up at all because they're thinner than a single pixel in this representation. And so you can see that one phylum of protostomes, the arthropods, is by far the largest protostome phylum, but it's also the largest animal phylum, and more than half of all of the animals, of all the animal species, are in fact arthropods. This is one of the phyla that we'll be talking about later. We'll also be talking about the mollusks, another very large protist group, protostome group. We'll also be talking about the nematodes and the flatworms, right? So the non-protostome phyla are all grayed out. So that's just this region down here. And the chordates and the echinoderms are the deuterostomes that we'll be talking about later. What I like about this particular representation, though, is it does give you an idea of how large some of these groups are in terms of the different kinds of animals. If we look at a different kind of representation, then a phylogenetic tree, which of course has no information about how many species, but just the relatedness of the different groups, then we can see that protostomes, as shown here in figure 34-2 from your textbook, are divided into two groups. One group is the Lophotrochozoa, and the other is the Ecdysozoa. And we'll be looking at just a couple of representatives from each group. But before we talk about the Lophotrochozoa, let's consider just one interesting point about something that has convergently happened in a number of different protostome lineages, as shown in figure 34-3, which is in front of you right now, which is a very common transition to a terrestrial existence from an aquatic existence. So that's shown in this figure by either green dots or green solid, depending if all of the members of the descendant lineage are terrestrial or just some of them. And so notice that in all of these cases, they are being derived from what were aquatic ancestral lineages, and it is the descendant lineages that have taken up a terrestrial existence. Now, there's an exact parallel with some of the issues that we talked about considering the early transition of plants from an aquatic to a terrestrial existence, and so it should not surprise you to learn what they are. One is waterproofing. As soon as you leave the water and you're out in the air, you're starting to lose water evaporatively, so desiccation is a real problem. So in any of the terrestrial groups, you see a lot of adaptations for minimizing water loss, such as making the outside more waterproof or diminishing the parts of the body that are in fact permeable to water. There's also issues with sexual reproduction. And so just as with the plants, the very first terrestrial plants were still completely dependent on films of water for the sperm to swim to the eggs. And similarly in the animals, you see that the terrestrial lineages have to do something quite different with, in terms of sperm transfer, so sexual reproduction in, I'll put in terrestrial up here. So sperm transfer can no longer rely on water in the environment, right? Not like the sponges that can just simply release the sperm into the surrounding seawater and maybe it'll hit the right sponge downstream or not. But so you get internal fertilization is much more common in terrestrial organisms because it's direct sperm transfer instead of just releasing sperm into the environment. And then another issue is changes in mechanical support, just as we discussed for the plants. Coming out of an aquatic environment where you're being buoyed up by the greater density of water, once you're surrounded by air, then you no longer have the mechanical support of the surrounding water. So huge changes then in mechanical support that's needed for terrestrial organisms. Okay, let's have a quick overview of the Lophotrochozoans before we consider two of the phyla for highlighting. So the Lophotrochozoa is this group right here. What a mouthful this name is, right? I think it's a little easier to wrap your mind around it if you recognize the different parts. And there are, in fact, 3 different characteristics that are found in some of the Lophotrochozoans. Confusingly, to students at least, not all Lophotrochozoans have all 3 of these characteristics, but many of them have at least 2. So let's go ahead and quickly go through these. So the lopho, the first part of this name, refers to a special structure called a lophophore, and this is a structure shown here in figure 34-2, and it is a filter feeding organ. And so where would you expect to see this? In organisms that are aquatic, because all the filter feeders or suspension feeders that you see that make their living as animals, pulling organic particles out of the environment, are all aquatic. And so this structure, the lophophore, is found in some, but certainly not all of these animals. But that's where the lopho comes from, so that's number 1. Number 2 relates to the trocho part of the name, and this is a characteristic larval type that is found, again, in not all of the Lophotrochozoans, but a number of them, and it is a larva that looks exactly like this. It's pointy on both ends, has a little girdle of cilia around its equator equivalent, and then a little tuft on one or both ends as well. It swims around. These are all aquatic. So these are only found in the Lophotrochozoans that have an aquatic larval stage. So this was something that was noticed by early zoologists, which allowed them to understand, prior to all the kinds of molecular characters that are now used today to corroborate those earlier phylogenetic trees, but it was existence of characters such as the trochophore larvae that allowed individuals scientists to recognize the relatedness between the annelids, the mollusks, and the rotifers, and the flatworms, and so on. And number 3. Here is number 3, and it didn't actually make it into the name, right? We covered the lopho, and we covered the trocho. What's the zoa part? Well, of course, that just means animal, right? So fortunately for us, it's not the Lophotrochospirozoa, and it would be called that if, in fact, it included all the different 3 major characteristics, because characteristic number 3 is a special kind of cleavage referred to as spiral cleavage. Now, cleavage, as you may recall, is the special name given to the earliest divisions in embryology. And in the Lophotrochozoans, then as the cells divide, they also twist such that the daughter cells are not lined up as they are shown down here on the bottom, which is found in some organisms, but they tend to twist like a sort of spirally sort of pattern as shown here, which is why it's referred to as spiral cleavage. So those are the 3 characteristics. Well, At this point, I would love to show you my cool flatworm movies, but we ran out of time, so I'll have to start with cool flatworm movies then on Friday, and we'll go through some of the phyla. This is where I stopped at 1 o'clock, so you get a break of a few minutes. I'll see you on Friday.